So, welcome back to our symposium, Accelerated Domestication of Orphan Crops and Wild Plants. This is a symposium on the very special circumstances. We have in the audience here in the Royal Academy of Sciences and Letters in Copenhagen, Denmark, we have, as I counted now, 9 people in the audience here listening to the talks. But the whole symposium, because of the circumstances, is live streamed. In fact, we told all people who registered for this symposium, and I can say it was fully booked a few days after it was announced the first time, without making any big announcements. So the interest is very high. But all people who signed up for the symposium have now got a link so they can follow the live streaming in real time. Yeah? And we have speakers from all over the world. Some of them are here. Yeah? We have brave people who went here, or maybe they regret it when they came here. But we have also people from abroad, and we will hear now 3 Americans, yeah, that were so wise that they decided to stay home. Yeah? But we will hope we can hear their talk anyway. Yeah? So the first speaker will be Professor John Dobley from the University of Madison, Wisconsin. Are you there, John?
Do you hear me?
Yeah, very excellent. Welcome. I'm just saying we are very happy that John can present here at our symposium, because he laid the whole theoretical background for domestication by his studies on maize genetics. So we are so happy that you are here today and that you can participate even in this very special way here. Yeah, but we're happy it works. So please go ahead, John.
Okay, thank you very much. Ordinarily, I'd say happy to be here, but I'm not there, so I'm just at home in my office where I normally camp out. So I would begin with the first PowerPoint slide, if that could be projected. I see the screen is starting. Okay, good. And I was told that I'll need to sort of indicate to the projectionist when to advance. So what I intend to do is just to explain the kinds of studies I've been doing over the last, dare I say, 30-some years, and maybe we can have a discussion of if there is relevance in that to create the opportunity to create new crops. So I'll say a bit about the origin and history of maize next. The past research, where I spent most of my career trying to go from phenotype to getting at the underlying gene, next. And then I'm going to spend most of the talk on more recent study of the genetic architecture of teosinte and how it compares to the genetic architecture in maize. And that, for me, is very informative about what domestication does. Next. How did the genetic architecture constrain domestication? And next, yeah, how did it, domestication alter genetic architecture? I think there may be one more. Yeah, okay. So first, a bit about maize and teosinte. On the left side, you see a teosinte plant. It's highly branched. And the cartoon on the far left makes an important point, and that is that these branches have many little ears along them, and the tip of the branch has actually a tassel or male structure. And during domestication, as you see on the right is corn, the branching was largely eliminated. If you look at modern corn, the types that modern plant breeders use in the advanced countries. And the side branch has just a single ear, not many little ears, and the ear is on the tip of the branch. So you had to change from having a tassel or male structure at the tip of the branch to having a female structure or ear. I like to think of this as corn has something of a constitutive shade avoidance architecture. In wild plants, if you're out in the sun, you branch profusely to fill up the space. If you're shaded, you don't branch. So the teosinte would not branch if it was strongly shaded. And so corn has sort of become a plant that sort of is in a constitutive shade avoidance mode. Next. Even though the plant architecture is very strongly different, the ear architecture is even more different. On the left, the teosinte ear has only 2 rows of grains, which sort of are oriented to opposite sides of the ear. But most importantly, you don't even actually see the grain. The grain is locked inside these hard little cases. And where the corn ear on the right, the grains are naked on the surface of the cob. And all of the structures that make up the casing in teosinte that surrounds the kernel are in the center of the maize ear and form the cob. The way I like to say this, it's a bit misleading, is that maize domestication turned the ear inside out because you took the kernels on the inside and moved them to the outside, and you took the structures on the outside, the casings, and put them to the center. Okay, next. This is me as a young man quite a few years ago in Mexico collecting teosinte. I guess the point is that it's a very large plant like corn. They're tall. They can be 12 feet tall. And you can see the branches on the plants I'm holding. And then next to it is a patch of teosinte growing along the stream, just to show you it's a wild plant. It's part of the native vegetation in Mexico. Next. We did early sort of phylogenetic study. That's a phylogenetic tree for maize and its relatives. And the top part of the tree has all different varieties of maize that were representative of the diversity in the New World before European contact. And up at the top, you can see maize from the Andes Mountains, and then down towards the lower middle part of the tree, maize from the southwestern United States and even northwestern United States. The point, I don't have a pointer. I don't think you see my mouse pointer if I use it. But anyway, the point here is that all maize traces back to one origin, one particular point in time, so that there was a single domestication event. And the green branches represent the teosinte that is basal to maize, and therefore the one we think is ancestral to maize. We also did molecular dating using the microsatellites that were used to create this phylogenetic tree, and we could get a divergence time of about 9,000 years ago, which is pretty close to what the archaeological data would tell us. So maize was domesticated 10,000 years ago in Mexico. Next. And if you look, this is kind of a map of Mexico, the central southern part of it. All those green dots represent teosinte populations of the type of teosinte that's most like maize. The starred dots, the asterisks, they are the populations of this type of teosinte that are closest to maize in the phylogenetic tree. And so they all come from this one area, and we think that's likely the area that was the center for the domestication of maize. Next. If you go down there today, this is what you'll see. That's a hillside. It's a larger hill than you might think from the picture, but that's all teosinte growing all over that hill. If you go up to the top of that hill and look out across the landscape, you just see hill after hill after hill covered with teosinte. And so it was a tremendous resource for these ancient people, because each of those plants produces quite a bit of grain. And so they certainly had a natural resource that they wanted to take advantage of as a food. Next. The archaeological record's pretty informative too. These are ears from an archaeological site in southern Mexico. On the left side, the oldest years from that site are about 6,000 years before present. It's already corn, but it's very small, has only about 35 kernels on the cob. And within that site, over time, the ears get progressively larger till where you get the ones around 600 years ago, about the time of European contact, and they are fully the size of modern corn ears. And what's really going on here is that way back when, 6,000 years ago, a plant might have 20, 30, 40, 50 of the small ears, each with 35 kernels, and now it has one large ear, maybe with 400 or 500 kernels, and it's just a change for ease of harvestability. So instead of needing to go and pick 20 little ears off a plant, now you just pick one giant ear. Okay, next. The seminal sort of breakthrough in understanding maize domestication came from this famous individual, George Beadle, one gene, one enzyme. And Beadle did an interesting experiment. He crossed maize and teosinte. And if you go to the next, or just hit forward, we'll see, yeah, there you go. And got it F1. And he saw the F1s were fully fertile, indicating maize and teosinte were the same species. And he selfed the F1s to make F2s, and then he studied trait segregation in the F2. And he proposed that there may be a small number, his number was 5, of sort of Mendelian factors that played a large role in the domestication of maize from teosinte. Next. And my lab focused on trying to identify and see if we could genetically clone these Beadle factors, and we had some success at it. And we did this by QTL mapping. So the central part of this slide is a QTL map. Along the bottom, you see the chromosomes. Maize has 10 chromosomes, but there were only 4 chromosomes that had a QTL for this trait. And the trait is the formation of that fruit case surrounding the kernels in teosinte, but not surrounding the kernels in maize. And so we just quantified that. Y-axis is the LOD score. It tells you its statistical significance. And we found one very large peak. In a sense, it behaves almost like a Mendelian gene. In fact, you can find genetic backgrounds where it will behave like a Mendelian gene. We named it teosinte glume architecture because it's the gene controlling the formation of the fruit case, which is composed of a botanical structure called the glume. Next. When we cloned it, we found out it was a SPB transcription factor. And quite interestingly, we were able to show that the difference between maize and teosinte was a single amino acid change, which altered the function of the protein, turning it likely, we think, from an activator into a repressor of its target genes. Next. We did this introgression experiment. So there on the left, you see a standard teosinte fruit case. And on the right, that fruit case has the maize allele of this teosinte glume architecture locus introgressed into it by back-cross breeding. And what it does is it blocks the complete formation of the fruit case, so the kernels become exposed. So this would have been a tremendous advance for maize breeding, because no longer did you need to get the kernel out of the teosinte casing. It would be there more or less open on the surface of a teosinte ear. Next. And we did some molecular work to show that many of the targets of this TGA gene were Madsbox genes, which was sort of expected. So we found the gene, we know its targets, and we think that what it's doing is regulating the formation of this fruit case. Okay, next. Here's another trait. So that little cartoon shows teosinte. You have many little ears on the side branch. On maize, you have just a single ear on the side branch. We were able to clone the gene for this. It was also a transcription factor. Here, the causative site was a regulatory gene or a cis regulatory element, CRE. It was about 7.5 kb, 5' of the open reading frame. Next. And what this did, this cis-regulatory element changed the expression pattern of the gene. These are tissue in situ hybridizations. They're cross-sections through a lateral branch. So you can see all of the kind of leaves coming off that branch. This is a very immature lateral branch. And where the leaves are attached, that would be what will become the nodes in the fully elongated branch. On the right side, as the arrows point, there's like a very faint band of expression at each node, and that's Maize. So in Maize, there's a faint band of expression at each node that's lacking in teosinte, which you see on the left side. And so what we think is happening is a gain of expression of this transcription factors in the node. And when you gain that expression, it's blocking the formation of or outgrowth of the lateral buds. So you do not get the little ears at each node along the branch. Rather, you only get a single ear at the top. And again, here we can see an introgression where on the left side, you see a single Maize ear on the tip of a branch. But if you introgress the teosinte version of this QTL into the Maize genetic background, then you get these multiple little ears along the branch, as well as the large ear at the tip. Okay, next. Finally, the last trait we'll look at for major genes is the overall plant architecture or branching of the plant, whether you have a very profusely branched plant like teosinte or a more or less unbranched plant like Maize. Next. We clone this gene. It's another transcription factor and the gene is teosinte branched 1. And you see its expression pattern in Maize. Those are tissue in situ hybridization through very small Maize seedlings. And the arrows point to the axillary buds. And the gene TB1 is expressed in the axillary bud where it represses the outgrowth of the bud, and therefore you do not get the branching. Next. And so what happened in domestication is that the Maize allele is expressed at a higher level than the teosinte allele. The higher level of expression causes greater repression of the branching, so you get an unbranched plant. And we were able to find part of the answer to how that happened, and it was the insertion of a hopscotch transposable element into the regulatory region of this gene. And that hopscotch element appears to act as an enhancer, causing the higher expression. Okay, next. So to summarize this part of the talk, we have been able to clone 5 of these major genes. All 5 were transcription factors. Some cases, it's regulatory changes are responsible for the difference between corn and teosinte. In one case, it's an amino acid change. Okay, next. We did some other work to see how these genes all work together. And we found lots of interaction, either at the statistical level or at the molecular level, that these genes all work in sort of a common developmental pathway. Again, to go back to what I said earlier, we think it's related to the shade avoidance pathway. So you see the phytochrome B up at the top does repress teosinte branched. And then teosinte branched controls a number of other genes, giving whether you have a branched or unbranched plant. Curiously, we found that the TB1 gene also regulates the fruitcase gene. Don't have time to go into all of that today. And then the fruitcase gene controls a series of Madsbach's transcription factors. I guess the point here is that there is this sort of molecular developmental pathway, which likely pre-existed in teosinte and was manipulated to get a different phenotypic outcome that is cultivated corn. Okay, next. Now, I don't want to leave the impression that it's all a bunch of big genes of large effect that one can get down to by QTL mapping. Here's another trait, kernel weight. Do you have small kernels or large kernels as in Maize? And here we've detected 21 QTLs. They have a range of effects from small to large. The LOD scores are not nearly as big as in my other examples. They're 40 as opposed to 150. But still, we can explain much of the variance. In fact, the statistical model, QTL model, explains the full heritability of the traits, 71% versus 72% for the heritability. A point I like to make about this slide, it doesn't actually, even though we sort of talk about it as if there were 21 genes, I think that's a misrepresentation of what it is. And way I like to think of it, it is a graphical representation of the best fit statistical model for explaining the maximum amount of variation in the mapping population. We've done simulations in our lab, and even though you fit 21, if there were actually 300 QTL, you would fit 21 QTL. 21 is sort of a sweet spot for the statistical model because of the amount of linkage in the population. So large numbers of QTL will only be detectable by a maximum number of 21. So, okay, next. Okay, I'm gonna go off to the second part of the talk. And this is CJ Yang. He was a grad student in my lab, and he's really the force behind this. He did all of the hard work, and in particular, all the data analysis to do this project. And it's a classical quantitative genetics analysis of maize and teosinte. Next. And what we tried to do is define the genetic architecture of teosinte, and we also defined genetic architecture of maize, and we used map populations. We went back to that area in Mexico where maize was domesticated, collected teosinte there, and from that same region, collected maize from the Mexican farmers living right there. Next. And so our questions are, how did domestication alter genetic architecture? Let's look at maize genetic architecture and teosinte and how do they differ? Next. And then how did genetic architecture constrain domestication? Are there things about how teosinte genes regulate development in teosinte that could have slowed down domestication? Next. Yeah. And the way we did this was just applying basically the logic of R.A. Fisher from a very long time ago on making estimates of genetic control of traits by looking at the correlation among relatives. Closely related relatives will be strongly correlated, more distantly related relatives less correlated. And using Fisher's model, one can actually learn a great deal about the genetic control of traits in a population. Next. And so we needed relatives. So we created a set of relatives by intercrossing 50 teosinte parents to create 4,000 progenies. And we did selfing and outcrossing, full sibs, half sibs, and some from two different parents would be unrelated individuals. So we've got this great mix of 4,000 different progeny. And we did the same thing with the maize landrace. This was done by Jim Holland, my partner in this project. He's a great plant breeder, and he's at North Carolina State. So then we had these two sets of 4,000 progeny, one for maize and one for teosinte. Next. We had to genotype them. So we did that by getting 40,000 SNPs on each of the progeny. So you see like a segment of chromosome there showing the location of each of the SNPs. Next. And we did whole genome sequence on the moms and dads for all of the progeny. And we could phase the moms and the dads. So we knew what SNPs were, alleles were on which of their two chromosomes. And then we could take the SNPs from the progeny and determine which of mom and dad's chromosomes they came from. So now you see the progeny SNPs are colored according to their origin. Next. And finally, we could take all of the SNPs from the moms and the dads, even ones never scored in the progeny, and project those onto the progeny. So we ended up with 18 million SNPs scored on each of the for 4,000 teosinte, 4,000 maize progeny. Next. And the mastermind, I call him mathemagician here, Peter Bradbury, worked out all the math to make this happen. And it was a scary process, but it seems to have worked. Next. We measured lots of phenotypes in the field. We grew our plants over 2 seasons. Next. And then we analyzed that data mathematically using a mixed linear model. In that model, we take all the trait measurements and we try to fit them to the additive or kinship matrix among the progeny, plus the dominant kinship matrix among the progeny. We put in a lot of other things into the model, field effects and so forth, the year it was grown. And out of that, what we get are estimates for additive effects on traits, dominance effects, the additive variance and the dominance variance. And importantly, we get a nice measurement of the narrow sense heritability, and the additive variance, which of course is the variance that plant breeders have to work with. You have genetic variance, which is composed of the additive and dominant, but the dominance variance does not respond to selection, only the additive variance. So this way we can kind of get a picture of how much additive variance there is in teosinte that could have been a target for the selective breeding of ancient agriculturalists. Next. Oh, that's Jim Holland, and Jim was the genius who fit all of these models and made it all work. Okay, next. So here's our first result, and this graph shows the total variance on the y-axis, different traits in the column graphs along the x-axis. Lower part are landrace results, and the upper part teosinte. The sort of salmon-colored parts of the graph are the additive variance, the blue, the dominant, and the green is the genotype by environmental interaction variance. Next. And I want to focus on this quadrant of the graph right here. And if you look at top at teosinte, you can see the orange segment, the additive variance, is very large. But in the landrace, the additive variance is depleted. And what we infer from this is that yes, there's a lot of additive variance for all these reproductive traits in teosinte, and it's been reduced through domestication. So as people selected to get larger grains and so forth, they were removing the additive variance from the population, bringing the good alleles to fixation, so you have much less additive variance, okay, in landrace. So there's the grain weight, for instance. There's a substantial difference between maize and teosinte in the heritability. Next. Yeah, if you go, okay, there's ears per plant, whether you have a lot of little ears along the side branch. There's a good amount of additive variance in teosinte, almost none in maize. Maize very rarely makes 2 ears on a branch. Next. Grains per ear. So teosinte has only 10 or 12, sometimes maybe 8 grains on an ear, but there's a lot of additive variance for that. Maize can vary from, 200 to 400 grains on an ear, but most of that's environmental. It's not due to additive variance in the population, so you see very little additive variance. Next. Yeah, and overall, if you just look at these different groups of traits, we divided our traits into 3 groups, vegetative and flowering time, and there maize has only about 80% of the variance of teosinte. Environmental response traits, these are like, does it tiller or not tiller? Are the multiple ears or single ears? There's been less of a loss there of additive variance for environmental response. But the real target where you've lost most of the additive variance are the reproductive traits. These are traits like grain size, grains per ear, and traits related to that. So there's been a real big decline. So with the decline in the additive variance, well, one of the things that happens is the dominance variance goes way up. I think there may be, if you go next, let's see, maybe a square. Yeah. So if we look at reproductive, The salmon color is the additive variance. The more bluish color is the dominance variance. And you can just see there's very little dominance in teosinte, but a lot in landrace. And the way we interpret this is as selective breeding removed the additive variance, brought the good alleles to fixation, which were left over with the dominant alleles, which could not be selected. And so dominance variance then becomes a larger part of the variance in the genetic variance in the population. Next. And then we just did this. We asked how much did the different traits diverge during domestication? And so it's rather simple. It's just the change in the mean phenotype in maize. This is on the y-axis. Over the mean phenotype in teosinte, how much did things change? And this is, go ahead next, yeah. So flowering time changed very little. The maize and teosinte were from the same area of Mexico, and maize flowers a little bit later, but not too much later. Next. The plant size changed, and so that maize is larger than teosinte, so there's a bit of change there, a little bit taller, broader leaves, and things of that nature. Next. You've got in maize fewer tillers, less branching, fewer ears per plant, so there's a decrease in there, so they're seeing a little more response now. Next. Grains per ear, so here you're getting a real gain in maize, very, very many grains per ear compared to teosinte. Next. Grain weight, much larger in maize than in teosinte. Next. And here's the shocker. Total grain weight per plant. How many pounds of meal or flour does a plant produce? And it's the same. And the way I interpret this is we really haven't improved yield in the absolute sense. What we've improved is harvestability. So instead of having 2,000 very tiny seeds on a plant, now you've got 200 much larger seeds. And so maybe perhaps the photosynthetic efficiency of the plant hasn't really been altered, but rather it's just been made easier to walk through the field, pick a single ear off of each plant. Next. Now, this is sort of a question for an evolutionary biologist, which is partly how I view the world. And so we want to ask, are all those differences in phenotype? We see maize has wider leaves than teosinte. Maybe that's just due to drift and not due to selection. So we wanted to do a test of selection and asked if the phenotypic divergence between maize and teosinte was a result of selection, or could it just be explained by genetic drift? Next. The way we do this is we take a statistic called FST, which is the proportion of the genetic variance at neutral markers between populations. Here the populations are maize and teosinte. How much of the variance between maize and teosinte is between populations versus within populations at neutral markers? Next. And you can do the same thing for quantitative phenotypic variation. How much of the phenotypic variation is between versus within the populations? That's called QST. Next. And you can make a graph like this. So the line there, the distribution line, shows the distribution of FST, and then you can plot on it where a QST for a particular trait would fall. And so if the QST value is outside of the norm of the distribution, then you have evidence for selection. Okay, so here's our results with teosinte. The gray shaded area is the distribution of FST values for neutral molecular markers. And then for each trait, the QST value is plotted. And basically what you can see, the most strongly selected traits, the ones far to the right, are all the reproductive traits. These are the things we think would have been primary targets of domestication. Our environmental response traits, which are in the salmon color, are also strongly selected, but not quite as much. And then the sort of plant size and flowering time are less selected, but the days to male shedding, which is DTA, and days to silking also still show evidence of selection, so that they have diverged by selection as opposed to drift. The one trait that shows no evidence of for selection, which you can see on the left, TGWP, that's total grain weight per plant. So it's again, total grain weight per plant didn't diverge, and a teosinte plant can produce as much grain as a maize plant. It's just going to be very hard to harvest. Okay, next. Yeah, okay, next. Okay, this is kind of another evolution question. How strong was selection? This is the breeder's equation, where the response is equal to the intensity of selection. Intensity of selection is the portion of the population that you are putting into the selected group versus the total population, times the heritability of the trait, times the amount of variance, the phenotypic standard deviation. Okay, next. So we want to get an estimate of that intensity of selection. That's just the normal distribution, and we measure intensity of selection in phenotypic standard deviation. So if you include 99% of the population in the selected group, only exclude 1%, then your intensity of selection would be 1%. Okay, next. So our estimates of the intensity of selection are really tiny. In other words, selection was not strong. That very, very tiny fraction of the plants each generation were excluded from contributing to the next generation. But if you keep that up for 10,000 years, you can make a lot of gain. Next. And just for comparison, this is from a study, Kingslover et al. On the x-axis, you have the selection intensity in a log 10 scale for natural species. And then if we go next, we can put on the teosinte values. So the vegetative traits are on the low end of what takes place in natural species, not very strong selection. Next. The environmental response traits, these are like branching and branch length and ears per plant. A little more strong selection there. Next. And these are the reproductive traits. Grain weight, grains per ear, things like that. Those are the most intensely selected, but there's still rather modest selection compared to what is seen in natural species. So the result or inference we make from this is that during domestication, there was not really strong selection. It was actually rather weak selection, but continued for 10,000 years. So that makes a lot of gain. And if we think back to those archaeological corn ears I've showed you earlier, they just change a little bit over that 6,000 year period for that figure. So for 6,000 years, each year just changing a tiny bit, you make a lot of gain over a long period of time. Next. Oh, and there's the one trait that doesn't show selection. It's the total grain weight per plant. Next. Now, I'm going to talk a bit about genetic correlations. So this is a way to think about it. In teosinte, you've got few seeds per plant. In maize, you've got a lot of seeds per plant. Teosinte has small seeds and maize has large seeds. And so in domestication, you wanted to do two things. You wanted to have bigger grains and more grains per ear. But whether you can get that or not depends on the genetic correlation between those two traits. Next. So let's say we wanted to do selection, and we want to get up to where the star is. And there's the distribution of phenotypes for those two traits in the population. Then selection would work quite well because the correlation between the traits is positive in a way that helps you move directly to your target. Next. However, if the correlation was like this, you'd have trouble. And that's because when you picked more grains per ear, you would actually end up getting smaller grains. And so you'd have a harder time getting to your target. Next. So what causes genetic correlation? It could be linkage or pleiotropy. And the consequences are it can cause a constraint, and it can also cause indirect selection. You select for one thing, but something else changes. Next. So again, if we have this optimum we're going for, if the correlation structure is as shown in that little white ellipse, then you'd have a problem. Next. And you can get to the optimum, but you have this sort of circuitous route, and so it takes longer. Next. You may have to go one more. Yeah, if the correlation structure is like that, you move much faster right towards the target. Yeah, you're good. Go ahead. So this is the genetic correlation matrix for maize and teosinte. Next. The top triangle is maize. The bottom triangle is teosinte. You'll see there's many fewer shaded squares in the top, and that's because in maize, things are less correlated. We think that's just if you remove a lot of additive variance, you have a harder time detecting correlations. In the bottom, teosinte has many more correlations because it has a lot more variance, so it's easier to detect the correlations. Next. For this part of the graph, in vegetative and flowering time traits, the correlations are pretty similar. In the little square, you'll see they're all blue in both above and below the diagonal. So the genetic correlations are very similar in maize and teosinte, stronger in teosinte. And the one drawn with a circle around it, again, you can see it's negatively correlated in both those two traits, but a little more strongly in teosinte. Next. This part of the graph, again, we see strong correlations in teosinte, a little less strong. This is the reproductive traits. But now we see some other kinds of interesting changes. Next. Here is an example where two traits are positively correlated in teosinte and negatively correlated in maize. And that's what you would expect if you've had strong selection. If this is traits, you're selecting out variants that have favorable positive correlations. As you remove those variants with favorable positive correlations, what's left behind are the variants with unfavorable correlations. And so you go from a positive correlation to a negative correlation. Next. And here's one, it's uncorrelated in teosinte, but becomes strongly negatively correlated, minus 7.75 in maize. So again, we're really changing the correlation structure for these reproductive traits. Next. So we wanted to ask how do the genetic correlations constrain domestication? So we started teosinte, we want to get up there to the optimum maize. Next. And the black line represents the trajectory of domestication. That's what actually happened. We went from teosinte to maize along that line. And we can measure the angle. Well, this would, where the theta value is the angle between what actually happened, which is the black line, and then the structure of genetic correlations in teosinte, which is shown as the red line. And this is, you got to remember, this is through a multi-dimensional space. We had 18 traits. So those are two vectors through a hyperspace of 18 dimensions. And we can measure the angle between them. If the angle is 90%, then you've got full constraint. Evolution will be very slow. Next. If the teosinte genetic correlations are perfectly aligned with what happened in evolution, then the angle would be 0 and you'd have no constraint. Next. You can go one more. So we looked at that, and initially the angle is 67 degrees. So there's a substantial amount of constraint that would have slowed down domestication because of unfavorable genetic correlations in teosinte. Next. And what you would expect is as you remove the favorable variants, with variants with favorable correlations, you should expect constraint to grow. And so if we look at evolution along the same path, and you are selecting in maize, there's even greater constraint. So we've sort of gotten up to an optimum. We've pulled out the easy selectable variants, and now it becomes much harder to make progress along the same trajectory. Next. So we can ask which traits cause constraint. Go ahead and click through this because I don't want to take up too much more time. Go ahead and, or just, I'll say something about the slide, but just go the next couple clicks. Yeah, so what this is trying to explain, there we go, we can get to this part, stop here. So what we do is we drop one trait. If when you drop a trait, the angle decreases, that means that trait was causing constraint. And what you can see is the angle decreases flowering time. So flowering time acted as a constraint to improve the other traits because it is unfavorably correlated with the things you want to improve, like grain size or grains per ear. So flowering time was imposed a constraint on improving along the trajectory of domestication. On the other hand, some of the traits like grains weight or ear diameter or ear length, they had positive correlations with the other traits. So those were the good traits, if you will, in that they worked well. So flowering time was basically the thing that would have slowed evolution down. Next. Let's see, this is the univariate breeder's equation. Next, which probably all of you know, I don't know. There's a multivariate equivalent. Next. And you take the genetic variance covariance matrix, you have a selection differential vector that you multiply times that, and you get the response vector, which is the change in phenotype for all the different traits. Here's it showing like 3 traits, so you've got Z1, Z2, Z3. And in matrix algebra, the equation is in the little block below where it's like delta Z vector is the G matrix times the beta vector. Okay, so next. Yeah, next. So we asked, the question here was which trait would be best to select on? Next. So we have these 18 traits, and you can ask which one, if I selected on it, would I make the most gain? And so the results are shown there. The black line is the true vector of domestication, how things actually went from the origin, teosinte, out to where maize is today. And if you selected on grains per ear, you would make more gain along the axis of domestication. And the angle between, the correlation angle between that trait and all the other traits is most favorable. And in general, the reproductive traits had the best genetic correlations, and the environmental response traits and the vegetative flowering traits the worst. So I guess the take-home message here is we can figure out which trait would have been most favorable to select, and it would have been the total number of grains per ear. So if you wanted to re-domesticate maize, go into a teosinte field and select the plants that have the most grains on each ear. Next. I will, just in the interest of time, I think I'll skip the next couple slides so you can. It's a rather complicated analysis, very cool analysis. Oh, stop when you get to the end of, or all the parts of this slide on, and I will make a comment. So go ahead, click a couple more clicks. Up, stop there. Yeah, so oops, you can go back one, sorry. What we did here is we looked at the structure of the genetic correlation matrix in maize and teosinte and asked how it changed. And for vegetative and flowering time traits, the correlation you can see there is 0.89. So the structure of genetic correlations for flowering time are the same in maize and teosinte. They're highly correlated. But so that means the underlying genetic architecture, the gene pathways, are still intact in the same way. But if you go down to the reproductive traits, maize and teosinte are uncorrelated. And what that tells us is that the genetic or molecular pathways underlying reproductive traits have been substantially rewired in the sense that genetic correlations are very different in teosinte from what they are in maize. So this is where change has really taken place, is on those reproductive traits. Next. This is Chuye Chen. She's my current postdoc. She mapped QTL using this population. So like for grain weight, she found 52 QTL. I would like to point out she found a very large effect. And go to the next slide. That's the arrow. And this is how big of an effect it is. If you look at the photograph of the teosinte grains, you'll see if you're homozygous for the one allele, you're rather small. Homozygous for the other allele, you're quite large. So there are some large effect factors out there in natural populations that could be tapped to advance domestication pretty rapidly. Next. And overall, what Chuye found was that the number of segregating QTLs in teosinte is much larger than maize. So the y-axis, number of QTL. If you just go all the way to the right, where it's all QTL over all types of traits, she found a total of 450 segregating QTL in teosinte, but only 213 in maize. So basically, you've removed a lot of the genetic variance in maize relative to teosinte. Okay, next. And this will be the last point. We could take the genetic variants, the heritable variants, the additive variants in teosinte and maize, and ask what portion of it can be attributed to QTL and what portion is unexplained. The unexplained is what is usually called in the literature the hidden heritability. And what we can see in teosinte, it's very much like humans. About 75% of the genetic variants we cannot map to QTL, despite considerable statistical power with our design. And so we think that that tells us, go ahead next, I think there'll be a little box pop up, that we think there are vast numbers of QTL of very, very tiny effect. And those are what are tremendous resource for plant breeding. You're not going to find them as QTL, but it's those things that are so tiny you can't find that are controlling most of the variation in teosinte and in maize. Next. Okay, this is the summary slide. You can just go through, and I won't say too much because I'm kind of out of time. But I'll just leave it with that question. Is there relevance for no-go crops? And thanks for listening, and I'll stop there.